Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
It's kind of wild to think about, but did you know Alzheimer's disease is actually the sixth leading cause of death in the U.S.?
Wow, really? I knew it was a big problem, but the sixth?
Yeah, it's the most common cause of dementia, too.
Makes sense. Those often go together.
But here's the thing. There's some really cool research happening right now that's, well, pretty hopeful.
Oh, really? What's that?
It actually connects back to Shakespeare, believe it or not.
Shakespeare? How so?
Or remember that line from Hamlet, "There's Rosemary, that's for remembrance."
Oh, yeah. Ophelia says that.
Turns out rosemary and its cousin, sage, they contain this compound called carnosic acid.
Oh, God.
And that's what's at the heart of some promising Alzheimer's research.
It's fascinating how this intuitive link between certain herbs and memory, something people have known for ages, is now being studied scientifically.
Right, like we're tapping into this ancient wisdom, but with modern tools.
And that's what we're diving into today, a potential new way to fight Alzheimer's, all thanks to this compound found in, well, our kitchens.
Exactly. Yeah. But there's a twist, right? Carnosic acid itself had some issues, but scientists have figured out a way to make it more stable, more usable.
Yeah, they've kind of tricked it out.
To break it all down for us, we've got two main sources: a scientific paper from the journal Antioxidants and a health news article from Scripps Research, both from March 2025.
So fresh off the press, then.
Yeah, pretty much. They both talk about these studies mainly done in the lab.
Right. Important to remember that it's early days.
Absolutely.
So what we're going to do in this deep dive is, go through the main points.
Lay it all out.
What is this new compound? How does it seem to work based on what they've found? What did the researchers actually see happening? And what could this mean down the road for treating Alzheimer's?
Trying to keep it simple, no jargon overload.
Exactly.
Okay, let's start with the OG compound, the carnosic acid.
Oh, gee, that's a good one.
So this isn't some made-up chemical, right? It's in rosemary and sage, totally natural.
Yep, you got it.
And it's already known to be kind of good for you, right?
That's right. Carnosic acid is a bit of a star in the world of antioxidants and anti-inflammatory agents.
Antioxidants, anti-inflammatory, remind us what those do again.
Basically, antioxidants protect your cells from damage.
Damage from what exactly?
Well, from things like pollution, stress, even just the normal processes of living can cause damage, and that's where antioxidants come in.
Like little bodyguards for our cells.
Exactly. And then anti-inflammatories help to reduce inflammation in the body, which is a natural process, but can get out of control sometimes.
Makes sense.
And the way carnosic acid seems to work is particularly cool. It activates these enzymes that are part of the body's own defense systems.
So it's like giving our natural defenses a power-up.
You got it.
But as with a lot of natural stuff, there's a catch, isn't there?
There always is, right?
I mean, it can't be as easy as just munching on rosemary.
Unfortunately not.
So what's the problem?
Well, pure carnosic acid, it's a bit finicky, it's really unstable, reacts easily with oxygen, breaks down fast.
Oh, so it doesn't last long.
Not really, which makes it a tough one to use as a medicine directly.
I get it, like it wouldn't store well and might not even make it through your system to do its job.
Exactly.
That's where the clever folks at Scripps Research came in.
Right. They thought, how can we get the benefits of carnosic acid without this instability problem?
And their solution was this new compound, DiAcCA.
Right. DiAcCA, exactly.
Break it down for us. What is that?
Okay, so DiAcCA stands for Di-Acetylated Carnosic Acid, and it's essentially a lab-made version of carnosic acid that's way more stable.
So they made it more robust, less likely to fall apart.
Exactly. And here's the really cool part. DiAcCA isn't actually the active ingredient itself. It's what's called a prodrug.
A prodrug. Sounds fancy.
Think of it like a disguise.
Carnosic acid in disguise.
Yeah. It stays stable in this disguised form until it hits your stomach.
And then what happens?
Then, bam, the disguise comes off, and you've got carnosic acid ready to be absorbed into the bloodstream.
So it's like a delivery system that protects the carnosic acid until it gets where it needs to be.
Exactly. Pretty clever.
Super clever. So they've got this stable version that turns into the good stuff once it's inside you. But the big question is, does it actually help with Alzheimer's? That's where the mouse study comes in, right?
Right. That's the next step.
So what was the main goal of this study? What were they trying to find out?
They wanted to see if DiAcCA could actually treat Alzheimer's disease in a living organism.
Okay, makes sense. But why mice?
Well, they use a specific type of mouse called 5 times familial Alzheimer's disease transgenic mice. which is a mouthful, I know.
5 times familial Alzheimer's disease transgenic mice.
Yeah, try saying that 5 times fast. But basically, these mice are genetically engineered to develop Alzheimer's-like symptoms much quicker than normal mice.
So it's like a fast-forward version of the disease.
Exactly. This allows researchers to see how a potential treatment might work over a shorter period.
Ah, so they're kind of like a model for Alzheimer's in humans.
Yeah, they develop key features of the disease, the amyloid plaques, the tau tangles we hear about, and even damage to the connections between brain cells, which is what ultimately leads to problems with thinking and memory.
So it's not just about those plaques and tangles, but also about how the brain cells are actually communicating with each other.
Right, that communication is key.
Okay, so they've got their special mice. How did they actually do the study? What did the treatment look like?
So they took these mice, about 5 months old, which is when they start showing those Alzheimer's-like symptoms,
Middle-aged mice.
and they gave them DiAcCA by mouth 3 times a week for 3 months.
Consistent dosing there.
And they used different amounts of DiAcCA, 10 milligrams per kilogram of body weight, 20 and 50.
Okay, so different dosages to see if there's a dose-response relationship, right? Like, does more drug equal more effect?
Exactly. And they had a control group, too.
Essential for comparison.
Right. Those mice just got olive oil, which is what the DiAcCA was dissolved in.
Ah, so that's their baseline, the group that doesn't get the actual treatment.
That's it.
Okay, so they've got their groups, their dosages, their time frame. Now, how did they measure whether the treatment was working? Like, what were they looking for?
Well, they looked at a whole bunch of stuff.
Thorough.
First, they did behavioral tests to see how well the mice could learn and remember things.
Like little mouse IQ tests?
Kinda. 2 important ones were the Morris Water Maze, which tests spatial learning and memory. Can the mouse remember where a platform is hidden in a pool of water?
Wow.
And then conditioned fear tests, which assess different types of memory formation, basically. Can the mouse remember that a certain cue is associated with something unpleasant?
Got it. So they're looking at different aspects of memory and learning?
Right.
But they didn't stop there, did they?
Nope. They also looked at the mice's brain tissue under a microscope, using special techniques to see things in detail.
Like a microscopic deep dive into the brain.
Yeah, they use these markers that can bind to specific things in the brain, like those amyloid plaques and tau tangles.
So they could actually visualize and count them?
Exactly.
What else were they looking at in the brain tissue?
They looked at the density of neurons, how many brain cells were there.
Makes sense. You want to see if brain cells are being lost or protected.
Right, and they also looked at the density of synapses.
Synapses, those are the connections between brain cells, right?
That's it. Those connections are crucial for communication between neurons, and they're often damaged in Alzheimer's.
Ah, so they're looking at how well the brain cells are talking to each other.
Exactly. And they also looked for signs of inflammation in the brain.
Inflammation, that's like the body's response to injury or irritation, right?
Yep. And it can happen in the brain too, especially in Alzheimer's.
So they were looking at whether DiAcCA could reduce that inflammation.
Exactly.
Wow, they really covered all their bases. So after all that, what did they find? What did the DiAcCA actually do in these mice?
Well, the results were pretty encouraging, I gotta say.
Let's hear it.
First off, they confirmed that the DiAcCA was successfully delivering carnosic acid to the brain.
Good, so their delivery system worked.
Yep, that was crucial. And then they saw that the treated mice, especially those getting the higher doses of DiAcCA, actually performed better in those memory tests, like the Morris Water Maze.
So their memory improved.
Yeah, they were better at remembering spatial locations. And in some of the tests, they were almost as good as healthy mice.
Wow, so the treatment seemed to be actually rescuing their memory function.
It really did. And remember those synapses, the connections between brain cells?
Yeah, crucial for brain communication.
Well, they found a significant increase in synapse density in the hippocampus of the treated mice.
The hippocampus, that's a key area for memory, right?
So it seems like DiAcCA was helping to either protect existing synapses or even promote the growth of new ones.
Like it was rebuilding those connections.
Exactly. And another big finding was that DiAcCA reduced inflammation in the brain.
That's what we were hoping for.
And what's cool is that it seemed to be more active in areas that were already showing signs of inflammation.
Ah, so it was targeting the problem areas specifically.
Right. Like a smart bomb for inflammation.
Clever. What about those amyloid plaques and tau tangles? Did they see any changes there?
Yeah, they actually observed a reduction in both amyloid plaques and tau tangles in the hippocampus of the treated mice.
So it was actually clearing out those hallmark Alzheimer's proteins.
It seemed to be.
That's huge.
And they also saw improvements in those brain immune cells, the astrocytes and microglia.
Less inflammation there, too.
Exactly. So it seems like DiAcCA was having a positive impact on the brain's immune response, calming things down.
This is all sounding really good.
It is. And remember, they also did that gene analysis, looking at which genes were turned on or off in the brain cells.
Right, like a peek into the inner workings of the cells.
Well, that analysis gave them some clues about how DiAcCA might be working at a molecular level.
What did they find?
They found that DiAcCA led to a decrease in the activity of genes associated with these things called disease-associated microglia, which are basically microglia that have gone rogue and are contributing to inflammation in Alzheimer's.
So DiAcCA was calming down those troublemaker microglia.
Exactly. And they also saw an increase in the activity of genes involved in protecting nerve cells, promoting the growth of new synapses, and even reducing brain cell death.
So it was like a multi-pronged attack on Alzheimer's, hitting it from all angles.
It really does seem that way.
And there was that other part of the study with human cells too, right?
Oh, yeah, that's right. They wanted to see if DiAcCA would have similar effects in human cells, not just mouse cells.
Makes sense. You want to see if it translates.
Exactly. So they took human microglia, which are those brain immune cells.
The same ones they were looking at in the mice.
Yep. But these were grown from human stem cells in a lab.
Ah, so they're like lab-grown human brain immune cells.
That's it. And they put these human microglia into mice that had been engineered to have human-like immune systems.
So they created a sort of hybrid mouse-human model.
Exactly. And what they found was that if they treated the human microglia with carnosic acid before putting them into the mice, it prevented inflammation that was triggered by those amyloid-beta and alpha-synuclein proteins.
Remind me what those proteins do again.
Well, amyloid-beta is the main component of those amyloid plaques we talked about, and alpha-synuclein is another protein that's involved in neurodegenerative diseases like Parkinson's.
Ah.
And what's really interesting is that carnosic acid was able to prevent inflammation even when antibodies to these proteins were present.
Antibodies, those are like the immune system's guided missiles, right?
Exactly. So this suggests that carnosic acid might be able to reduce inflammation even when the immune system is actively targeting these abnormal proteins, which is relevant to some current Alzheimer's treatments that use antibodies to clear out amyloid.
So it could potentially work alongside those existing treatments.
That's the idea.
Okay, last question about the study itself. You mentioned that DiAcCA was more stable than regular carnosic acid. Did they find any other advantages?
Yeah, they actually found that DiAcCA was absorbed into the bloodstream about 20% better than carnosic acid.
So it's more bioavailable.
Exactly. And it was, of course, confirmed to be more stable, which we already talked about. And importantly, the mice seemed to tolerate it really well.
No nasty side effects.
Not that they observed. In fact, some initial safety tests even suggested that DiAcCA might have a calming effect on inflammation in the esophagus.
The esophagus, that's the tube that connects your mouth to your stomach, right?
Yep. So it seems like DiAcCA might have some beneficial effects beyond the brain.
Wow, this is all sounding really promising. But of course, we need to be cautious about getting too excited. Let's shift gears now and talk about what these findings might actually mean for the future of Alzheimer's treatment. What are the implications here?
Well, the big takeaway is that DiAcCA is looking like a strong contender for a new Alzheimer's drug.
A potential game changer.
It's got that improved stability, good absorption, ability to reach the brain, and all those positive effects they saw in the mice.
It seems to be ticking all the boxes.
And what's really exciting is that it seems to be targeting multiple aspects of the disease at once.
Which is often the best approach for complex diseases, right?
Yeah.
Hit it from all sides.
Exactly. DiAcCA seems to be tackling inflammation, oxidative stress, synaptic loss, and the buildup of amyloid and tau.
So it's not a one-trick pony.
Nope. And we talked about the potential for it to be used in combination with those existing amyloid-clearing antibody treatments.
Right, like a tag team against Alzheimer's.
Exactly. The idea is that DiAcCA could help those treatments work even better and maybe even reduce some of their side effects.
Side effects like brain swelling and bleeding, right?
Yep, those are known as ARIA-E and ARIA-H, and they can be serious.
So DiAcCA could potentially make those treatments safer.
That's the hope. And another plus is that carnosic acid itself already has a good safety track record.
Right, it's on the FDA's generally regarded as safe list, right?
Yeah. Exactly. So that could potentially speed up the process of getting DiAcCA into human clinical trials.
Because regulators would be more comfortable knowing that the active form is generally considered safe.
Right.
It's like it's got a head start.
Exactly. Yeah.
Now, thinking beyond Alzheimer's, could this research have implications for other health problems?
That's a great question. And the answer is potentially yes.
Okay. How so?
Well, since DiAcCA seems to be so good at reducing inflammation, it could potentially be useful for other diseases that involve chronic inflammation.
Chronic inflammation, that's like long-term, ongoing inflammation, right?
Yep. And that's a factor in a lot of diseases like type 2 diabetes, heart disease, even other neurodegenerative diseases like Parkinson's.
So DiAcCA could potentially be a multi-purpose treatment.
That's what they're hoping to explore.
It's amazing to think that something derived from common herbs could have such wide-ranging effects.
It really is. But as with any exciting new research, it's important to be realistic.
Keep our feet on the ground.
Exactly. So let's talk about some of the limitations of this study.
Okay, what are the caveats here?
The biggest one is that this research was done in mice.
Mouse models, right?
Yep. And while mouse models are super valuable for studying diseases, they don't perfectly mimic what happens in humans.
So what works in mice doesn't always work in people.
Exactly. So we can't assume that DiAcCA will have the same effects in humans, even though the results in mice are very encouraging.
Got it. So we need to be cautiously optimistic.
Exactly. Another important limitation is that DiAcCA is still in the preclinical stage of development.
Preclinical, meaning it hasn't been tested in humans yet.
Right. It's shown great promise in the lab and in animal models, but there's a long way to go before it could become an actual treatment for people.
There are a lot of hurdles to clear.
Absolutely. And even though this study gave us some good insights into how DiAcCA seems to work,
Like activating that NRF2 pathway.
right, the nuclear factor erythroid 2-related factor 2 pathway, which is involved in protecting cells from damage.
A mouthful.
It is. But the point is, we still need to learn more about the precise mechanisms and the long-term effects of DiAcCA.
So more research is needed.
Definitely.
Okay. I always find it interesting to hear what the researchers themselves have to say about their work. What were some of the key takeaways from the scientists who led this study?
Well, Dr. Stuart Lipton, who's the senior author and also a clinical neurologist.
So he sees patients with Alzheimer's.
Right. He was really excited about the results.
What specifically stood out to him?
He emphasized that they saw an increase in synapses in the treated mice, which is huge.
More connections between brain cells, that's a good sign.
Definitely. And he also pointed out that DiAcCA helped to reduce the levels of those misfolded proteins, the phosphorylated tau and amyloid-beta.
So it was clearing out those harmful proteins.
Exactly. He was also optimistic about the potential for DiAcCA to be used alongside existing treatments, and he's hoping to fast-track clinical trials because of the good safety profile of carnosic acid.
So he's eager to move this forward.
Absolutely. And then Dr. Phil Barron, who collaborated on creating those stable carnosic acid derivatives.
He's the chemistry whiz.
Right. He talked about how they carefully selected DiAcCA as the most promising candidate because of its stability and how well it's absorbed.
So they put a lot of thought into choosing the best version.
They did.
It's inspiring to hear their passion and their hope for the future.
It is.
Well, as we wrap up this deep dive, what's a final thought you'd like to leave our listeners with?
I think this research really underscores the incredible potential that lies within natural compounds.
Like the stuff we find in our gardens and kitchens.
Exactly. People have been using plants for healing for centuries, and now we're starting to understand the science behind some of those traditional uses.
It's like we're rediscovering ancient wisdom through a modern lens.
That's a great way to put it. And it makes you wonder what other secrets are hidden in the natural world, just waiting to be uncovered.
What other potential cures are out there just waiting for us to find them?
Exactly. It's exciting to think about.
It really is. And for our listeners who are eager to learn more, where can they go from here?
Well, keep an eye out for news about any clinical trials related to DiAcCA that might start in the future.
Definitely. Those are always good to follow.
You can also explore research on the NRF2 pathway.
That pathway involved in protecting cells, right?
Yep. It plays a role in a lot of different diseases, so it's a hot area of research.
And for the real science buffs out there, they can always check out the original research paper in Antioxidants.
Exactly. It's got all the nitty-gritty details.
Awesome. Well, this has been a fascinating look into a potential new weapon against Alzheimer's disease.
It's a tough disease, but research like this gives us hope.
It does. And while these findings in mice are super encouraging, we need to remember that human trials are the next crucial step.
Absolutely. We need to see if DiAcCA can live up to its promise in people.
Fingers crossed. Thanks for joining us on this deep dive.
It's been a pleasure, as always.
And to all our listeners, stay curious.
And stay hopeful.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.